I'm here to talk to you about immunology. Immunology is the science that's important to our health as humans. In fact, it's important to the health of the entire world. So, just what is immunology? Immunology is a branch of science that explores the immune system and the diseases that can be found in it. We can learn a lot about the immune system from immunology. Immunology supports the understanding of how diseases work and how they're made inside of us. So, where did this amazing science start? Well, the concept of immunity, when the body can't get a specific disease, comes from all the way back to 5th century BC. That's a long time ago. Immunology really started to come about during the smallpox epidemic in China during the 10th century. Because of this epidemic, China came up with the concept of variolation, where scabs from the disease would be put on a non-infected individual, resulting in the non-infected individual having some sort of immunity to the disease. Many years later, a new idea was being worked on. This idea was called vaccination. Vaccination was thought to give people immunity, just like variolation. It involved putting a dead version of the disease into a healthy person's body in order to give them immunity. The foundation for vaccination was by Robert Koch, who found that bacteria could be transferred from animal to animal. Modern vaccination was then discovered by a man named Edward Jenner, who injected James Phipps with cowpox, a disease similar to smallpox, and then found that Phipps was then immune to smallpox. The inventor of modern vaccination could be named as Louis Pasteur. Pasteur left one flask of chicken cholera bacillus on his bench for an extended period of time, which weakened the virus. He then injected 8 chickens with the virus from the flask, and no disease was made. After vaccination, a new concept that's very important to immunology today came along. What was this concept? Phagocytosis, or when the immune system defends itself against a foreign threat. Phagocytosis was discovered in 1882 by Ilya Kmekenhov, who jabbed the thorn of a rose onto a starfish and noticed that soon after he did this, cells were surrounding the wound. Phagocytosis is caused by something called the immune system. The immune system is a large network of cells, tissues, and organs that defend the body against threats called antigens, such as viruses. It also defends against living organisms called microbes, such as protists and fungi. The way the immune system attacks foreign invaders is called the immune response, which we'll talk more about in detail later. Basically, all of immunology revolves around the immune system. The immune system is made of many parts. A main part is white blood cells. Just so you know for the rest of the video, white blood cells are also called leukocytes. White blood cells are found in clumps of lymphoid, leukocyte housing tissue in the body, called lymph nodes. They are also found in the spleen, thymus, and bone marrow. So how do these mini medicines get around the body? Lymphatic vessels and blood vessels circulate the leukocytes all around. By putting white blood cells all around the body like this, the body prepares for defense against disease and threats. White blood cells actually have 2 types, phagocytes and lymphocytes. Phagocytes are the white blood cells that attack foreign organisms. They're mainly made of cells named neutrophil, which are fighters of bacteria. Lymphocytes help the body to remember the organisms that have attacked it. They help the body defend against threatening organisms by providing a sort of source of memory. Lymphocytes come in 2 types as well, B and T lymphocytes, and T lymphocytes are also called T cells. B lymphocytes find foreign threats and send other cells to fight them, and T lymphocytes attack those foreign threats. While white blood cells are a large part of the immune system has many other parts to it. Among these are lymph, a liquid that circulates throughout the body and contains a high number of lymphocytes, lymph nodes, clumps of tissue that make lymphocytes and house them, the thymus, an organ that makes lymphocytes and matures T lymphocytes, the spleen, an organ that filters blood, provides a small supply of blood, disposes of broken down red blood cells, and has lymphatic tissue, and finally macrophages, cells that digest cellular debris and foreign substances. So earlier we mentioned the immune response. Well, what exactly is it and how does it work? Well, the immune response is a process that lets the immune system defend the body against foreign invaders, as we also mentioned earlier. Now, how does the immune system and response work? First, leukocytes are produced in the lymph nodes and other organs, and lymphocytes are produced in the lymph nodes. Then, possible threats are detected by B lymphocytes. Then, neutrophil, T lymphocytes, and many other cells attack the invader. This entire reaction is why you sometimes get fevers. The immune system is actually made of 2 different immune systems, innate and adaptive. The innate immune system basically works with cells that defend against bacterial infections. Its main goal is to defend against viruses and microbes. The adaptive immune system is made of antibodies. Antibodies are proteins made to defend against pathogens, which are viruses and bacteria. These antibodies work to defend against specific pathogens that the body has encountered before. This is how the body is able to attack pathogens that change over time. Just as there are 2 types of immune systems, there are also 2 types of immunity, innate and adaptive. Innate immunity is basically inherited immunity. Almost all humans have innate immunity. Innate immunity is a general defense against antigens. It uses the skin, chemicals in the blood, and a lot of cells that attack antigens. Adaptive immunity is very antigen-specific, meaning that instead of generally attacking antigens, it recognizes each one and attacks it differently. The adaptive immune system includes a memory, meaning that after an attack against an antigen, the next attack is much more efficient. The immune system also uses a crucial concept called immunological tolerance. Immunological tolerance is when the immune system doesn't respond to an invader that could trigger the immune response. It's basically the opposite of the immune response. There are 2 types of tolerance, peripheral and central. Peripheral tolerance makes sure that T and B lymphocytes coming from central tolerance do not cause an autoimmune disease, or an odd response of the immune system to a totally normal bodily organ. Peripheral tolerance uses clonal deletion and suppression. Clonal deletion is the process of which self-reactive T and B lymphocytes, the cells that cause autoimmune disease, are removed through a process called apoptosis, or programmed cell death. Clonal suppression is carried out by TREGs, suppressor T lymphocytes. Suppression is when TREGs slow down or suppress the breeding of self-reactive T or B cells. Then there's central tolerance. It's the main mode of tolerance. Central tolerance is the process where newly made T and B lymphocytes are made to be non-reactive to themselves, so they don't cause autoimmune disease. It happens when immature T and B lymphocytes have their cellular processes completely reconstructed. Just like science itself, immunology has branches. One branch of this amazing science is viral immunology. Viral immunology is immunology that involves viruses, how they work, and what the body does to protect against them. Main parts of viral immunology include the humoral response, neutralization, and viruses in general, or how the body responds to them. One of the main parts of viral immunology is viruses. Viruses are immunogenic, meaning that they are able to produce an immune response from whatever organism they live in. Viruses are always evolving and developing new strategies against the responses of their host bodies. There are many different classifications and two types of viruses. Classifications include bacteriophage, animal, plant, and fungal, and the two types are DNA and RNA. DNA viruses are divided into double and single-stranded DNA viruses, and RNA viruses are divided into positive and negative sense RNA viruses. Viruses cause two types of immune responses, humoral and cellular, which we'll discuss soon. As for bodily responses to viruses, our bodies produce antibodies, proteins made to defend against pathogens, viruses, and bacteria in response to viruses. Remember when we discussed vaccinations earlier? Vaccinations help to produce more antibodies, cause immunity, and assist with the humoral and cellular responses. Eukaryotes, creatures with cells with nuclei, including plants, perform RNA interference in addition to those other bodily responses. RNA interference is where enzymes in the organism detect a certain type of double-stranded RNA molecule and then destroy any single-stranded RNA molecules of this type. A main response against viruses is the humoral response, which involves neutralization. A second main response is the cellular response, which involves T lymphocytes projecting tiny fragments of their proteins onto the cell's surface, resulting in the destroying of a host cell if fragments of viruses are found in the proteins. Remember when we mentioned neutralization? Well, neutralization is the main part in the humoral response. Neutralization is the process that some antibodies use to battle viruses, and its definition is the blocking of sites which viruses use to enter the cell. Neutralization makes the viruses less pathogenic, easily spread, and infectious. Alright, so immunology is full of branches, right? Well, we're going to discuss another huge one. Immunomics. Immunomics is the use of methods that use entire genomes to study the regulation of the immune system and its response to pathogens. Its goal is to study the immune system as a working network, rather than in segregated pieces. Now, before we start, you're going to need to know what a genome is. What's a genome? A genome is a set of all of an organism's DNA. This includes all genes as well. Also, you're going to need to know about genomics. What's genomics? Genomics is the study of the genome in an organism. So just where did immunomics come from? Immunomics is actually a pretty new branch of immunology, so it doesn't have a significant amount of history known about it. In fact, it's so new that it can give us a really good insight into what new technologies for immunology could look like in the future. But that's off topic. What is known about immunomics is that Elizda and his team discovered that the use of cDNA microarrays, which we'll talk a little bit about later, could find clear gene expression in immune cells. So they essentially created immunomics. This same team created the lymphochip, a small cDNA microarray which contains over 13,000 genes that simulate the genome of a lymphocyte. We'll talk more about this too. Also, Iyer and his team in 1999 conducted studies which warranted the further study of the immune system as a whole and as a network, rather than in individual parts. What exactly does immunomics do for us? For starters, it lets us improve our current understanding of the immune system. It lets the immune system be studied as a whole and as a complex model, instead of in individual parts. It also helps scientists to better understand the regulation of the immune system and how it functions. Immunomics also shows previously unseen characteristics of the immune system, such as its cells constantly being in motion and its cells easily being able to change their shape. Secondly, immunomics can help detect immune system defects. The genomic technologies and insights of immunomics can help detect immunodeficiency, the inability of the immune system to protect the body from infection and disease, autoimmune diseases, diseases such as type 1 diabetes and arthritis, and tumors, bunches of cells that don't perform normal cell function, consume a regular amount of resources and can lead to cancer, and that's just to name a few. We also mentioned cDNA microarrays earlier. Just what are those? Let's break the word down. cDNA is DNA created from reverse transcription. Reverse transcription is the generation of cDNA from reaction involving single-stranded mRNA templates in reverse transcriptase. cDNA is used to make proteins express genes that they would not normally express. So, that's cDNA. What's a microarray? Microarrays are two-dimensional arrangements, like this square here, of large amounts of biological material that assay the material. P.S. To assay is to quantitatively measure or quantitatively assess a target entity, or if you want to be fancy, analyte. There are also different types of microarrays, like peptide and antibody microarrays. We also mentioned lymphochips earlier. What are those? Lymphochips are cDNA microarrays that are enhanced for genes relating to the function of the immune system. They are made specifically for the purpose of studying the immune system. So, what did we learn about immunology today? Immunology is a branch of science which explores the immune system and the diseases which can be found in it. It teaches us a lot about the immune system. Louis Pasteur, Edward Jenner, and Robert Koch all helped to found our current understanding of immunology. Phagocytosis is when the immune system defends itself against a foreign threat. It was discovered by Ilya Mekhanov. The immune system is a large network of cells, tissues, and organs that defend the body against threats called antigens. White blood cells, or leukocytes, are a huge part of the immune system and have two types, phagocytes and lymphocytes. Lymphocytes help the body to remember the organisms that have attacked it and come in B and T lymphocytes. The immune response is a process that lets the immune system defend the body against foreign invaders. The immune system is made of two different immune systems, innate and adaptive. There are two types of immunity, innate and adaptive as well. Antibodies are proteins made to defend against pathogens, viruses, and bacteria. Immunological tolerance is when the immune system doesn't respond to an invader that could trigger the immune response. There are two types of tolerance, peripheral and central. Tolerance is generally meant to prevent autoimmune disease. Viral immunology is a branch of immunology that involves viruses, how they work, and what the body does to protect against them. Viruses are immunogenic and cause the humoral and cellular responses. Vaccinations help to produce more antibodies and cause immunity. RNA interference is where enzymes in the organism detect a certain type of double-stranded RNA molecule and then destroy any single-stranded RNA molecules of this type. The cellular response involves T lymphocytes projecting fragments of their proteins onto the cell surface, resulting in the destroying of a host cell if fragments of viruses are found in the proteins. Neutralization is the process that some antibodies use to battle viruses, and its definition is the blocking of sites which viruses use to enter the cell. It's the main part of the humoral response. Immunomics is the use of methods that use entire genomes to study the regulation of the immune system and its response to pathogens. Its goal is to study the immune system as a working network rather than in segregated pieces. Immunomics lets us improve our current understanding of the immune system. cDNA microarrays and lymphochips are important parts of immunomics. Well, that's all for today. See you next time.